I'm Sam, and I'm not a physicist. And I'm here with Eric, who is a physicist. We're the B-Brains. And today we're talking about the scientific method. Now, recently we've been talking about it in physics, but now, today, we're talking about how that compares to the scientific method in biology. So, Eric, take us away.
Yeah. Hi, Sam. Uh, right. My training was originally in physics. I have a PhD in physics, specifically material science. But then over the years, more and more of my research was in biology, in plant developmental biology, in plant and animal cell biology. And I want to talk about that transition because the differences between the way physicists do science and the way biologists do science are pretty profound. And I don't if you're not in the trenches of scientific research, I don't think you're likely to appreciate these differences. First, all right, so I want to start with a story from really one of my first serious lab experiences in biology. Okay, so first let's start with what I was used to. So what I was used to in physics is that reproducibility was easy in the sense that any physicist who wants to study an electron anywhere in the world is studying the same thing, right? And in fact, it's a fundamental principle in physics that every electron in the universe is literally identical to every other electron in the universe. So much so that if you put two electrons in a box and you don't observe them, and then you look back a little bit later, you can't tell which electron was which. In fact, there's a joke among physicists that there's actually only one electron in the universe, but it just keeps zipping back and forth through time over and over again. So we keep re-experiencing the same single electron. Uh, Sam, have you heard any of this before? Or
I have heard that about electrons. I haven't heard that joke, but we should have an
oh
episode
yeah,
where we just talk about jokes in physics and
oh
philosophy.
yeah, yeah. Um, right. So the point is that reproducibility in physics is easy in the sense that two different labs trying to do the same experiment on an electron should get exactly the same results. There's no problem there. So physics has there. I mean, like if I were to ask you, do you think physics is harder, or if you ask an outsider, do you think physics is a harder science or biology is a harder science? Sam, if I asked you that question, what would you answer?
Oh, I mean, physics seems
Right, and
true.
why
Math is
is
harder.
what is your that right? I think that's most people's
Math is
gut
harder.
reaction. And what's your reason for that statement?
That the math is harder?
Yeah, right. So physics is famous because for having incredibly difficult mathematics, and that is true. Um, I mean, I taught physics for years, and I saw some students successfully manage the math, and other students left physics not because they loved physics, but the math was just too much of a challenge for them. And yeah, and so for instance, my PhD thesis was a 30-page paper that was almost nothing but advanced mathematics, right? And
Wow.
that's completely typical for physics. But it turns out that if you take the math away, if you take math out as a consideration, physics is much easier than biology, which sounds like an extraordinary claim. But so let me tell you this little story that might illustrate why biology can be more challenging than physics. So it was around 2005. I was doing I had a year off from teaching, and I spent it in the lab of Tobias Baskin at UMass Amherst. And that was a plant biology lab. And one of the things that I was doing was I was growing plants. Now, I was growing, there was a little weed that we worked with called Arabidopsis thaliana. And it's a common laboratory plant because it's tiny. You can even grow the seedlings in a Petri dish. You don't need a lot of soil. And one other nice thing about it is that because of the biology of Arabidopsis, you can make thousands or tens of thousands of Arabidopsis seeds, which are all genetic clones of one another. So they're all genetically identical. And this is what I was doing. I was taking these little seeds and I was growing them in Petri dishes. So you could actually see both the stems and the roots because the growth medium was transparent. And what I noticed is that after a few days the seeds would germinate and the roots would start growing into the growth medium. And I noticed that every single root was different. I mean, if you've ever looked at plants for even a moment, then you that's not a surprise, right? I mean, the root system, just like the branches of a tree, they branch at unexpected places. And so just the same as no two elm trees are the same, no two Arabidopsis root systems were the same. And so but this, as a physicist, this was weird for me. So I spoke to my colleague Tobias, and I said, now Tobias, by the way, he has a background in physics, but he spent his whole career in biology. And anyways, but I said to him, look, these seeds, they're all genetically identical. So why aren't the root systems all identical too? Why does every root system branch in different places? His first reaction was to kind of laugh at this question because no biologist would ever ask this question. Biologists simply assume that genetic clones still aren't identical in the same way that no two identical twins are ever truly identical. But Tobias had to unpack the reasons for me. So he pointed out that although the genetics might be the same, every seed is slightly different, right? Because the seeds grew inside a seed pod in the maternal plant, and each seed in that seed pod would have gotten slightly different amounts of sugar and stored up slightly different amounts of starch. Maybe the growth hormones they were exposed to were different in small but relevant ways. When one cell multiplies into a small lump of cells, in this case, a plant embryo, there are regularities to that process, but no two sets of cell divisions are ever going to be truly identical. So at the end of the day, when the seed germinates and the root emerges, even though each seed is genetically identical, each seed is still going to be physiologically different. Different amounts of hormones, different amounts of sugar and starch, different amounts of protein availability. And there are I mean, in biology, there are actually other additional problems or differences that one might encounter. You'll notice I instantly wanted to call these problems because as a physicist, I know that what you want are things which are truly identical and things which are reproducible between labs, like electrons, because without that, how are you ever going to communicate? So Sam, how are we doing so far?
Oh, I'm following. I'm fascinated. This is cool stuff.
Yeah. So, are you beginning to see the outlines of why I'm going to argue that if you ignore the math side of things, biology can be harder than physics?
Well, I was thinking it's a messier system. It's not as uh, clean cut. Yeah, more
Yeah. Yeah.
variables, I guess,
Say
and
that again?
more variables where you don't even know what they are.
Right. Right. Yeah. So exactly right. Yeah. So if you were to ask me, well, how are these two seeds different? And then therefore, how are their root systems going to be different? I mean, I could take the two seeds, break them down, measure the amounts of protein, measure the amounts of hormones. Having broken down the seeds, I couldn't then germinate them, but I could come up with some hypothesis for why certain things influence the branching pattern in certain ways. I mean, it would be a 5 to 10 year long project, and at the end, I may or may not come up with the ability to predict a little bit about the branching patterns. But that's just within one lab. Okay, so within one lab, we have genetically identical seeds, but they're each physiologically different. And then we're growing them all on the same exact growth medium, which is to say, it's a mix of nutrients and something solid like literally jello so that the plants have a solid surface to grow on. When you put some sugar in there and whatever other nutrients the plants need to grow without too much of a leaf system to get them started. And then on top of that, there's also something called the light regime and so we actually grow these plants not in a greenhouse, but in a light box where we time the amount of dark and light. Basically the length of a day might be exactly 8 hours or exactly 16 hours. And we also know exactly how much sunlight or a simulated sunlight these seeds are getting in a day. And so now, if we were to compare our results to the results of another lab somewhere, who knows what, like that other lab, they, even if they are also working with the same clone of seeds that we're working with, which is a pretty common thing, actually, uh, you can still have to ask, what is their growth medium? Did they, because you would think that every lab would more or less standardize their growth medium so that two experiments could be compared. But it turns out that's definitely not the case. Tobias mostly liked to study roots. And so he had settled on a variant of the standard growth medium that made the roots especially healthy. Okay, but every scientist who studies plants is interested in perhaps a slightly different aspect of their growth. And so it's not unusual for them to adjust the growth medium a little bit to favor root growth or favor leaves, or maybe they want to study the effects of phosphorus. So they prepare a version of the growth medium that doesn't have enough phosphorus so they can add it in as part of the experiment. But you can see where this is going. Or worst of all, maybe they grow their plants in a greenhouse. And so suddenly it depends on how many cloudy days there were in Denmark in 1984. So pretty soon the question of, if I do an experiment on Arabidopsis, and I'm interested in the root growth, and someone else does an experiment on Arabidopsis, and they're using a greenhouse in Denmark. I mean, to what extent are those experiments going to be comparable at all? From a physics point of view, remember, we're used to studying electrons or something similar, which are literally identical no matter where you are in the universe and at any time. So in biology, we have to worry about how no two seeds are identical, no two laboratory settings are identical, no two growth media are identical. If you're working with greenhouses, and if you're working with large plants, you need a greenhouse. You have to worry about things like that. Pretty soon you start to wonder whether biologists can do science at all. Uh, it's
Okay, so let me ask a question here. Someone who
Yeah.
doesn't know much about this. Um, would some biologists say that even though they don't have exactly the same setup, the features of the plants that they're studying are indifferent to the differences in the laboratory setup? So sure, there are differences, but those differences don't really matter for what we're studying.
Well, yes. So that is the hope, right? So what biology experiments hope to find are what you might call universals. I mean, honestly, physicists are looking for universals too, but it turns out that finding universals in physics is much easier than finding universals in biology, right? So a universal effect would be, let's say you knock out a specific gene in Arabidopsis. Okay, for example, there's a gene called PIN2, and if you break that particular gene, then the roots no longer have the no they can't sense gravity anymore. The roots just tend to grow out sideways rather than digging down into the soil. And it turns out that no matter whether you're growing your plants in a greenhouse or in a light box, whether you're in Denmark or the United States, the roots just lose the ability to detect gravity. So that's an example of a universal. And so if we found in biology, if we found a universal, then that's great. And it means that we can move forward and begin asking questions about, why is that gene relevant for gravity sensing? And what kind of protein does that gene code for? And are there other genes that are related to that function? So biologists, the good news is that in biology, such universals do exist. And if you're lucky enough to find one, multiple groups can sort of hang their hat on the same coat rack, if I can stretch an analogy or metaphor. Um, okay, so yeah, so how we doing, Sam?
Yeah, well, I think we're doing good. Yeah.
Okay, but the challenge is finding those universals. Okay, so um, I'm curious if you do you have any idea how do you have any idea how biologists would go about looking for a plant that can't sense gravity? if all you have to start with are plants that do sense gravity, do you have any did your biology course in college ever teach you anything about that?
Oh, yeah, I don't think there was a biology course in college. So,
Oh yeah,
um,
okay.
yeah, so they're
Um,
looking for a plant that can, I mean, is it like a water plant or something like that?
oh, interesting. So you do comparative biology. Yeah, actually, that's actually an excellent suggestion. Right? So you look at a plant that grows in an environment where gravity matters less, and I think that's a really good suggestion. And I actually love comparative biology, and I've in my career I've done a fair amount of it. But it turns out that in modern biology, comparative biology is it it's not where most of the important work is done. And the reason is, oh boy. All right. So why do 100 groups all over the world study Arabidopsis when Arabidopsis is not a commercially important species? In biology, there's a ten, or here, actually, there's something more familiar. Why do groups all over the world who are doing cancer biology tend to study white mice rather than cancer in humans, or even cancer in monkeys for that matter? Do you have any thoughts on that, Sam?
I mean, ethics. I would assume the ethical
Well,
challenges,
right. I mean, I'm not
but
suggesting
also
that
it's
we
just
do.
higher
Yeah.
populations. Is it like
But in other
easier to
words,
keep them?
so it turns out that in biology, for better or worse, every species is different, and it's hard to know. It's hard to make a list. Actually, never mind that. It's impossible to make a list of all the ways in which two species might be different, right? So let's say you wanted to study, study mice and compare them to some other species of mouse that is less familiar, that only can be found in Australia or something, right? So you could imagine doing a comparative study where you compare the standard lab mouse and the Australian mouse and see how they respond differently to, for instance, a cancer drug. But that's virtually never done. And it's not done because, and more generally, comparative biology is not widely done in the modern world because it's in a sense it's already too hard to understand what's happening inside laboratory mice, full stop. So if you have 50 groups studying white mice and 50 white lab mice and 50 groups studying these Australian mice, then at some level those two groups of scientists are going to find it very difficult to ever have a conversation because they're not even working with genetic clones. So instead, it's tradition in biology, and it's been the case, actually, it began in the 20th century with fruit flies. Biologists discovered that if they wanted to have anything meaningful to say to one another, they needed to all agree ahead of time that they were going to study a single organism or a single cat, so they were all going to study white lab mice, or they were all going to study Arabidopsis thaliana, right? If you're studying Arabidopsis thaliana and you start talking to someone who studies rice plants, you can have a conversation, but those conversations are going to be hampered in many, many ways. Actually, this is a critique of, for instance, biology, because although Arabidopsis is easy to study, obviously the things that are relevant for human industry and human health would be the food crops, wheat and corn and rice. And so I mean, maybe 10, 15 years ago, there was a growing pressure in funding agencies to force, for example, plant biologists to begin transitioning the information they'd gathered by studying these tiny weeds, Arabidopsis, into crop plants that were economically relevant and relevant for human health and well-being. But as a result, comparative biology is very few people do comparative biology. It's just there's a most biology works with a handful of model species, and that when you make an important universal discovery in a model species, you then cross your fingers and hope that that discovery will translate into the species you're actually interested in. So if you discover a cancer gene in mice, then begins the process of trying to confirm whether it also is true in primates and ultimately true in humans. Um, So again, these are the challenges that biologists face that physicists who study fundamental particles absolutely never need to wonder about. So am I beginning to convince you that if you don't have to worry about mathematics, that biology might be more challenging than physics?
Sure.
Okay. Um, let's see. Now, yeah, this is as good as okay. So these statements that I've been making about my own experience, for example, uh, what I haven't told you yet is that, right, so if you're lucky enough to find a universal effect, then that's great. And so, right, this pin2 gene, if you break it, then Arabidopsis plants lose their ability to sense gravity. And it turns out that there are similar families of genes that exist in rice and wheat and corn. And I don't know if we've checked in all these plants yet, but I do know that if you break these genes in crop plants, you can get maybe not identical effects, but similar effects. So there are some very reproducible and universal effects in certain gene families. And so if you're lucky, you discover such a gene, and then you can spend 10 or 15 years studying it, and then try to do the translational research to move your discovery into an economically important crop. All right. So Sam, but now this brings us to the fact that how many discoveries that you make in a lab are universal versus how many discoveries that you make in a lab turn out not to translate between, say, your lab and Denmark, or between a growth chamber and a greenhouse. So, Sam, this is as good a place as any. Do you have the paragraph handy that I mentioned?
Oh, sure.
Okay, so I'm going to ask you to read a paragraph from a paper that was published in 2012, and it has to do with something called the reproducibility crisis in biomedical science. So go ahead.
Okay. This is Mobley et al. To examine a microcosm of the academic experience with data reproducibility, we surveyed the faculty and trainees at MD Anderson Cancer Center using an anonymous computerized questionnaire. We sought to ascertain the frequency and potential causes of non-reproducible data. We found that 50% of respondents had experienced at least one episode of the inability to reproduce published data. Many who pursued this issue with the original authors were never able to identify the reason for the lack of reproducibility. Some were even met with a less than collegial interaction.
Collegial.
Um, Conclusions, these results suggest that the problem of data reproducibility is real. Biomedical science needs to establish processes to decrease the problem and adjudicate discrepancies in findings when they are discovered.
Right. Okay, so let's recap. So this was a relatively early paper in on the topic. This is at a cancer center in Houston, Texas. And there was, I guess, this paper, if I had to guess, this paper emerged from a set of conversations around like a lunch table where maybe someone who's interested in the sociology of science, or maybe just a scientist who's interested in the process of science, started talking to his colleagues or her colleagues and discovered that that at least half of them had tried to reproduce somebody else's research, not necessarily still in Houston, maybe a Paris research or a London research, but that at least half of all the people at the table had tried reproducing someone else's work and failed. And so they put out an anonymous computerized survey for all the people in the cancer center. And I don't know what the response rate was, but when the responses rolled in, what they discovered is that half of all respondents had at least tried to reproduce someone else's results and failed. And that when they did try to dig into the results, sometimes the response, they couldn't figure out why why their attempt at reproduction had not succeeded. And furthermore, there was that snide comment about collegiality, which is to say they were told to they were not treated kindly. Or they were ignored or worse. And does this surprise you, Sam?
Oh, well, I mean, it's like 50% of people had that experience, but I wonder of the people who had the experience, how many times did they have the experience, or how many times, I mean, I guess I could go read the paper, but how many times did they like reproduce it and succeed? I feel like I want to know the base rate here.
Oh, yeah, no, I mean, I think what it's saying is, I don't think it's saying that 50% of the time they tried to reproduce it. I think that among the people who tried to reproduce it, oh wait, no, it's 50%, yeah. Right. I mean, so you can dig into the details of the survey. This survey was followed up by several others, and the bottom and across the board. And again, I should emphasize this is not really a problem in physics, but it's a problem in biology, in medical research. It's even a bigger problem in psychology, but we're not going to talk about psychology. But the fact is that when you do anonymous surveys of scientists, what you find is routinely people try to reproduce the results of reported by other labs, and failure rates range between 25, 30%, 50% sounds a little high for these surveys, but this is pretty standard that uh, just that many scientists have encountered this. The thing is that usually it doesn't end up in the literature, right? So you have to do these anonymous surveys because very few scientists are willing to go on the record and say, I tried to reproduce this other person's result and I couldn't reproduce it, right? It's because scientists are human beings. And if you go on record saying that you could not reproduce their work, one of you, the implication is that one of you did something wrong. And it's, it's just scientists are people, and it's a way to generate anger and animosity, and you have to work with these people. You have to go to conferences and be civil with them. The next time you apply for a government grant, they might be on the panel that's deciding whether you get money for your research or not. I mean, it's a no-win scenario. Okay,
Yeah.
there's virtually no good way to publish a paper saying we tried to reproduce this result and we failed, right? Because by implication, either you aren't as good a scientist as the other group, or the other group did something wrong. I mean, there's no good outcome there.
Yeah.
Um,
So I've heard of maybe similar challenges which have to do with publishability,
yeah.
that
Oh,
it
publishability. Okay.
yeah, that like null results aren't published very much
Yeah.
because the journals won't want to do it. But it is important. It's an important scientific result, and people don't experience that. And also sometimes that there isn't attempts at reproducibility for expensive experiments because you can't get the funding to do it, and no one wants to spend money on something that someone else has already gotten the glory for publishing. So I feel that I've heard of those two dynamics happening in
Yeah,
science.
that's exactly right. I mean, it's not an official part of the scientific method, right? In the scientific method, it's very inward looking. So you make a set of measurements, you come up with a hypothesis, you do a controlled experiment, you process the results, you refine your hypothesis, and then you do your next experiment. The way science works in reality is you're part of a larger community. For example, all the people working on Arabidopsis plants, or all the people working on clones of white laboratory mice. And so your experiments have to fit into a larger patchwork of other people's experiments, and hopefully you're friendly with these people and you see them routinely. But yeah, if someone makes an important discovery in biology and publishes a paper on it, and then you set out to reproduce their result, even if you do confirm the result, it's very hard to get that paper published because it's just confirmatory. And if you don't manage to confirm their result, and you can't figure out why, it's also hard to get your paper published. I should say that this was a set of conversations that broke into the literature in the early teens, right? So about 10 years ago, 15 years ago, there were a lot of conversations around this uh, defect in the scientific method, which is to say that reproducibility wasn't as widespread. Reproducible attempts at reproducing results were not as widespread as they should be, and conversations about reproducibility were not as common as they should be. So coming out of this, there were I mean, there were attempts to change the situation. So, in particular, there are now things called data repositories, which is if I do an experiment to check someone else's work, I don't need to write a paper. I can just, or I don't need to publish a paper. I can just process my results, write a little paragraph explaining them, and just submit them to an online archive that for free, and where anyone else can access the data. And that way I don't have to, I'm not picking a fight with another lab. I'm just saying, here's my experiment, here are the results. Anyone who wants access to these results in the future can have access to these results for free. So that was one attempt at solving the problem. Another attempt at solving the problem was there were one or two journals which were created to be sort of low cost avenues of publishing, low cost avenues for publishing experimental results in which you are trying to reproduce someone else's work. And then there was actually in psychology, which again, it's not our main focus today, but in psychology, I believe there were one or two large scale attempts at organizing a set of reproducibility trials where psychologists intentionally tried to reproduce each other's work. So these conversations all came to maturity around 10 years ago, and there were some productive outcomes. I mean, I would say that systemically science still suffers, the biological and medical sciences still do suffer from these problems, but at least now many people in those fields know that there is such a problem.
Yeah. Do you feel like people are maybe using machine learning to
Uh-oh,
survey those kind of archives to see what the status is of a certain experiment? Yeah.
interesting.
How many people have, attempted to reproduce this result?
Oh boy. Right. So now you get to the question. So I believe between the lines, the question is, if someone got famous by publishing a paper, do they care if other people aren't able to reproduce it?
Oh, I wasn't, that was not in my brain when I asked the question.
Okay. So okay. See, so how would you phrase it?
Well, I was just I'm just thinking of like there's so much going on. There's this expansion of the numbers of journals, the number of evidence and data. And so if you're a working scientist and I'm relying on this experimental method, and this is the way that doing the protein thing, and that you rely on so many different methods. And if every day more and more people are uploading their archive on these archive sites,
Yeah.
their data or their attempt to do it, I feel like there needs to be a machine learning solution so that I can easily scan that data and see if there have been any issues with any of the methods that I'm using.
Okay.
So I'm
So
yeah.
okay. So I could right. With a little luck, I would say 10 years in the future from now, when AI knows what science is. For the record, I'm not quite sure. AI doesn't quite know what science is yet, but it's getting there. 10 years from now, when AI knows what science is, maybe it will be able to provide some insights into the reproducibility crisis. I don't think it's there yet. I mean, I could be wrong, but I don't think, I don't think that there's enough I don't think there's enough of a archive of attempts at reproduction to draw any widespread conclusions. And circling back around to my point, I think that if there's a scientist who got a job and maybe tenure by publishing a series of papers that made them famous, the fact that no one else can reproduce the results is the last thing they want to worry about. I think I wish I could tell you that this was rare. I'm not sure. I'm not sure it is rare. It's been my experience that many of the most famous scientists are also the ones who care least about reproducibility.
No.
Actually, no, that's an exaggeration. I would say some of the most famous scientists are the ones who care least about
Yeah.
reproducibility. Um,
Now, here is an interesting distinction between philosophers and scientists.
okay.
So, you're pointing to a certain kind of scientist who wants there to be nothing but acceptance of their theories. But among philosophers, if you make an argument and there's no refutation of it, it just kind of dies on the vine.
Oh, okay.
And
Interesting.
we would often suspect that there is a little bit of, I don't know if it's an explicit agreement, but some kind of implicit agreement between like two people. Like, okay, I'm going to make an argument and publish it. I get bashing you. Go ahead and make an argument bashing me back and publish that. And then I'll publish. And so you're both stacking up the publications by criticizing each other.
Interesting.
So which is good. You have more
Yeah.
publications.
Well, like I said, in biology, if you're lucky, you make a discovery, and then that discovery turns out to be universal. So, for instance, this PIN2 gene discovery did turn out to be universal. And so in that case, other groups, like so, for instance, another group could take your discovery as a starting point, and then they could discover a gene that interacts with your gene, and they could become famous by publishing, their discovery that EIN1 interacts with PIN2, and that they both shape how a plant detects gravity, or for that matter, detects sunlight or some such thing. So I mean, there's certainly plenty of good science in biology where you make a discovery that does translate between labs and which is reproducible, and where just multiple groups start exploring interlocking pieces of the same phenomenon. So that's the ideal, and it does happen. It does happen a lot. But there are, so in my own experience with plant biology in particular, I behind the scenes, I had a chance to see both kinds of discoveries, right? So I saw and spoke with biologists who made these universal discoveries, and then they became famous, but their discovery was ultimately reproduced in other labs, and other labs did related work, and everything went smoothly. But equally well, I'd encountered, I think actually, I will say this is probably the minority of people, but there are people who publish high visibility papers on a topic, and but then other groups can't reproduce the results, or even worse, other groups come up with reasons why that discovery may not have interpreted their experiment correctly. But once someone has tenure, it's very hard to dislodge them from an opinion, because you can't, they won't be fired. And if someone has enough power inside a field, they can kind of coast. I've seen this. I've seen it more than once. So that would be a person who, again, they're philosophically, they're just not that worried about reproducibility, and they're just going to keep chugging along. There should be, there should be mechanisms, there should be more self-correcting mechanisms in science, but in some cases, those mechanisms break, especially when you get someone who's both powerful and tenured. It's a challenge. Yeah. So I think do we have time for me to tell one more anecdote?
Yeah, I think that'd be
Okay.
great.
Okay. So, right. So I was a physicist by training, and I moved into biology starting around the year 2000, maybe a little earlier. And then I spent about 15 years mostly doing plant biology, plant developmental biology, plant cell biology. Excuse me. I worked with a little bit with other systems. And but because I was a physicist, I was always interested in the raw data. I was always interested in the numbers, in the spreadsheets, in the statistics. I always had more of an interest in the math than most biologists typically do. So if I was interested in a topic, I would typically email the person who had published the paper, and I would say, "Hey, can I have your raw data, or can I have your Excel spreadsheets where you calculated this result?" Now, sometimes I'd get ignored, but I would say maybe a third or half the time, the person who published their result would send me their raw data. There are other instances where there's some branches of biology now where it's completely standard to put all of your raw data in an archive. And so in that case, I didn't need anyone's permission. I could just go to the free archive and download the raw data myself. So so over the course of about 15 years, I probably let's say I probably double checked let's call it somewhere between 30 and 50 other scientific papers. And I would say that among those double checks, I would say I found at least one small mathematical mistake. Oh, maybe a quarter of the time or a third of the time. And I would say that I found what I would describe as fairly profound misrepresentations of the data twice.
Hmm.
Okay. So
Sounds
let's
like
see.
you
So
should
what
be
is
a
that? So
reviewer.
what was that?
Sounds like you should have been a reviewer more often.
By the way, what I'm describing to you, this is not the way to rise to the top of a field. Uh,
It's the way to make friends and influence people.
yeah. Right. So like if you discover what you think might be a problem with someone's analysis and you email them out of the blue, uh, yeah, I mean,
I mean, it worked for Socrates.
actually,
I mean,
for what it's
he
worth,
got killed for it, but now he's famous.
yeah,
That was his
that's right. That's
general
right.
strategy.
Socrates is
I'm
a
going to
lesson about
poke a hole in, in everyone's theory.
Socrates is a less is an object lesson in what happens when you try to peer review the work of someone powerful. Um, yeah, so, but just to recap, so about 25% of the time there was at least one small mistake. And then let's be generous, 2 out of 50 or 4% of the time, there was what I would call a really substantial error in interpretation. Um, so that's my own personal experience. And I will say, actually, when I found a mistake, especially in cases where someone had been generous enough to send me their raw data, what I would do is I would email them back and I would say, look, here's the mistake I found. You don't have to worry. I'm never going to tell anyone about it. I leave it up to you in terms of how you want to respond to it. Maybe the next time you write a paper, you include a footnote that includes a correction. Maybe you never mention it to anyone. It's entirely up to you. Because I Partly because I knew that if I went public with the mistakes that I'd found, no one would ever send me the raw data again. I would just
That
be
is true.
right. I mean, no one would ever want to take the gamble that a mistake was lurking in their calculations. So I was always very clear that if someone was willing to share their raw data with me, that I would never publicize a mistake, and I would never even write a paper based on their raw data without double checking that it was okay with them first. I mean, on the flip side, one time someone shared their raw data with me, and I analyzed it, and I came up with a new conclusion, and so I wrote my own paper on it, it was part of a larger work, and I emailed them. I'm like, look, can I include your data? And they're like, oh, sure, just go ahead. So that's the way science is supposed to work, but then again, in that situation, I hadn't found a mistake. Maybe I was quibbling over a point of interpretation, but I weren't saying they had done anything wrong. Just the opposite. I was saying the result was so interesting that you could build an additional piece of science on it. So that's the ideal
Yeah.
situation.
Did were any of the major misrepresentations people who'd sent you the data?
Yeah, actually, yeah. I mean, I don't want to get into I can't for obvious reasons I can't get into any of the details. But
Right.
yeah, so
Did you
there
email them
wasn't
about that?
there was at least one instance where someone sent me their raw data, and I really differed over how they'd interpreted the data, um, and I told them as much. And they continued to be very friendly with me, but at the same time they weren't they didn't they just felt that I was wrong, and I no matter what I did, I couldn't convince them that I was right.
Hmm.
Um, and so I mean, I wouldn't call that a worst case scenario because at least they were still friendly to me. Uh, but that's certainly not the ideal outcome in science.
So if you could do one thing to address this issue of reproducibility in biology, what
Yeah.
would it be?
Oh boy. Well, actually, it is. I think this is something that's already starting to happen. Yeah, journals are changing their publication policies now. Putting raw data in public archives is now becoming a standard part of scientific publication. So that's the one thing I would have changed, and that actually is happening. So I
Great.
guess this conversation does have kind of a happy outcome here, which is there was a problem that was recognized in around 2015, and over the subsequent 10 years, scientific journals have taken a lot of steps to improve the situation.
Okay, great. What's your next plan? What's stage two?
Um,
Eric's plan to, improve biology.
yeah. Well, maybe we can leave that for another podcast.
Okay, that sounds perfect.
Yeah.
Well,
Oh,
you've
okay.
been,
So right. So I'm sorry. Here's our outro. Is I'm Eric and you're Sam, and we have been the Boltzmann Brains. Thanks for listening.
thank you very much.